structure of DNA. This is the phosphorus, the ribose sugar, and a base, and this is called a nucleotide. The bases are held by covalent bonds, hydrogen bonds, sorry. Weak hydrogen bonds.
Damn, look at you, yellow.
And these are called covalent bonds, which holds a new ladder with another ladder of bases of DNA. And a phosphorus, a sugar, and a base is called a nucleotide. So this is our prokaryotic and eukaryotic DNA. A prokaryotic is one circular strand of DNA and only has one replication fork, which is this whole thing. Has two replication forks, but has one origin of replication, so it's just a continuous circle of DNA. And our eukaryotic DNA has multiple replication forks, which means that this could be a replication fork, this whole thing could be a replication fork, and so can this whole thing be a replication fork. And it is a linear, unlike the prokaryotic, which is a circular DNA. Now moving on to our RNA and DNA. RNA has the bases, guanine, cytosine, uracil, and adenine instead of thymine that matches with adenine. It has one strand of DNA and has multiple structures such as mRNA, tRNA, and rRNA. However, DNA has two strands, has bases of guanine, cytosine, thymine, and adenine, and these two still match together. And it stores genetic information. Unlike the RNA, which stores protein structure information. Lastly, we talk about Rosalind Franklin. She confirms the DNA's double helix structure and was the foundation for Watson and Crick's DNA structure, which they stole from.
Okay, so now we're going to talk about DNA replication. So DNA replication occurs in the DNA, in the nucleus. So in order for replication to be successful, there's 6 crucial enzymes that are part of DNA replication. So first, we start with helicase, which unwinds a single strand of DNA. It unwinds it into 2 strands and it creates this replication fork. If you can see here, is like it's still like the DNA strand still together, and then here is unwind, so helicase would be here moving this way. So replication goes from 5 to 3 prime, and for that reason DNA replication occurs like in opposite directions in each strand because they're anti-parallel. So now we have DNA gyrase, which helps relieve tension at the replication fork, which is here because of everything that's occurring here. It helps this be able to continue to separate into 2 strands. Then we have DNA primase, which lays down the primer. A primer is necessary for DNA polymerase 3 to add nucleotides because it is unable to start on its own. Then DNA ligase comes in and it forms bonds between the nucleotides because there are something called Okazaki fragments, which are in sections, and DNA ligase helps put them together to make one solid line. And then comes DNA polymerase 1, which removes the primers, the mRNA primers, and transforms them into DNA. The whole process of replication is semi-conservative because half of a mother strand is kept and then the other is a daughter strand, so there is always going to be one original piece of genetic material.
to begin the process of transcription, an RNA polymerase binds to a promoter sequence of a gene so that it may be expressed. And to enhance the expression of a gene, an activator will bind to an enhancer to increase the rate of transcription. And on the other hand, a suppressor will bind to a silencer to decrease the rate of transcription. So mRNA, at one end of the mRNA is a poly A tail, and at the other end is a 5' cap. And so what they both do is they help to protect the RNA from degradation. So there are 3 types of RNAs. There's messenger RNA, transfer RNA, and ribosomal RNA. So mRNA for short carries genetic codes from the nucleus to ribosomes. Transfer RNA decodes mRNA sequences into a protein, and rRNA binds to accessory molecules for protein synthesis.
Okay, so now we're gonna go over translation. So translation is basically the process that uses an mRNA copy to build proteins out of amino acids. So how exactly does this begin? So we start with codons. They have to be in pairs of 3. So then this is a pair, this is a pair, and this is a pair. Keep in mind that everything in translation occurs within the ribosome. A ribosome will carry a strand from 5' to 3'. So let's begin the process. AUC will translate into an anticodon, which will become UAG. AAA will become UUU, and then ACG will become UGC, which is basically making the codons to anticodons. These codons will later on be labeled as amino acids. So basically the anticodons that are pairing with the codons are the complementary base pairing, which is gonna keep going until each of these finds a matching codon and anticodon within the ribosome strand. So then after this, each ribosome will shift to read the next codon as it continues to go. So everything will be moving from 3' to 5' in this direction as it keeps on pairing. And then afterwards, the ribosome will continue to move on, and then it will add each amino acid. So each single one of these paired with these will continue to go on until the strand has been completed, and then this is what determines what the final strand will look like.